My name is Kara Lucas, this is Jennifer Jang, and that's Natasha Garamani, and we did our project on cell division in microgravity. Cell division is important to life because all organisms must go through the cell cycle in order to grow, develop, and reproduce. Past experiments that have been conducted on Earth and simulated microgravity have shown that a lack of gravity causes a slower rate of cell division. The purpose of our experiment was to discover the effect of microgravity on cell division and to compare the rates of cell division in the absence and presence of gravity. Our hypothesis was, if cells are exposed to microgravity during their life cycles, then there will be a reduction in cell division because of the disorganization of microtubules, which are necessary for the formation of the mitotic spindle in cell division, the change in actin filaments, which affects the formation of the cleavage furrow in cytokinesis, the chromatin becoming less dense, which affects the formation of the chromosome, and increased apoptosis, or programmed cell death.
Some of the materials that we used were fibroblasts, which are skin cells that assist in the healing of wounds, Eagle's minimum essential medium, which helps us maintain a healthy condition of cells, 1% fetal bovine serum, which contains nutrients, a light microscope to observe attachment, a hemocytometer to count the cells, pipettes to transfer materials, and trypan blue to stain. To prepare for our experiment, we first coated the inside of our flask with 3 milliliters of polylysine solution, which helps with attachment, and we let that sit for 5 minutes. Afterwards, we aspirated the solution out and let it dry overnight. The following day, we added 500 fibroblasts to each of the flasks, as well as 5 milliliters of Eagle's minimum essential medium with 1% fetal bovine serum. We then allowed both flasks to incubate overnight. The following day, we added 6.3 milliliters of Eagle's minimum essential medium with 1% fetal bovine serum to each of the flasks. We kept one in the incubator and allowed another one to stay in a controlled room temperature environment. We allowed both flasks to incubate for 12 days so that they can grow and divide.
Then we had to count the cells. So first we washed the cells with 2 milliliters of phosphate buffered saline solution, and then we added 0.5 milliliters of trypsin or EDTA and rotated the cells for 1 minute. This way we could allow for the dissociation of the cells from the bottom of the flask. Then we added another milliliter of phosphate buffered saline solution. After that, we removed the medium from the flask and placed it in a 15 milliliter centrifuge tube and centrifuged it on a tabletop centrifuge for 5 minutes to pellet the cells and get rid of the liquids. And then we took out the cells and resuspended it in another 0.5 milliliters of phosphate buffered saline solution. Then we stained the culture with trypan blue and then counted it with the hemocytometer. So to do this, we added 10 microliters of the cells into the hemocytometer and placed it under the light microscope. Because they've been dyed with trypan blue, they're now very clear under the microscope, and so we used the nice grids out of the hemocytometer to count it. And how many cells there were per grid, we multiplied that number by 104, and that was the total number of cells per milliliter that we got. We counted it twice to make sure it was quite accurate. So this was our ultimate data. Our initial count for the first flask that we plated was 500 cells per flask, and we actually ended up with an average count of 482.5 cells. However, when we plated it at 750 cells per flask, instead we ended up with an average of 1,272 cells, a huge increase. This was probably because since there were more cells in the second sample with 750 cells per flask, they were able to come into contact more, which allowed for more cell division and also helped the cells to attach. Since fibroblasts need attachment to survive, more cells were able to survive and grow and reproduce. Then we placed another flask in the incubator, and this one was plated at 500 cells per flask, and we ended up with an average count of 1,616.5 cells, another great increase. And these are just some of the pictures of us performing our experiment.
After the experiment ended, we concluded that growth is more prominent for fibroblasts in an incubator at 37 degrees Celsius rather than in room temperature because the incubator mimics body temperature, which is more favorable for cell growth and division.
We would like to thank Mr. Wyeth Colo for being our advisor, Dr. Susan Kane and Ms. Erin Denny of City of Hope for providing the lab space and materials, the Smithsonian Air and Space Museum for hosting this event, and SSEP for providing this invaluable experience. Thank you.
All right, thank you for wonderful presentations. Do we have questions for our researchers? Please come to the microphone.
Why did you use Tri-Band blue?
Oh, we used trypan blue so we could dye the cells. This way, when we looked at it under the microscope, we could count it more easily instead of having to count the white cells because they're really clear and hard to count.
Okay.
Hi, I was just wondering, did you make this experiment as trying to solve some harms in society, or was it just basically out of curiosity?
It was mostly out of curiosity because fibroblasts are essential in the healing of wounds, so we were wondering whether or not, say, they would heal faster or slower in space, things like that. So it was mainly out of curiosity.
Thank you again.
Thank you.